Now we are going to start about major functions of natural killer cells. First, starting about the things we have to know about their key roles. First, they kill human cells infected by viruses. And second, it produces interferon gamma to activate macrophages. Secondly, it is a member of innate immunity. It's a member of innate immunity but is origin in lymphocyte. It is the only one cell of lymphocytic origin that belongs to innate immunity. And they're all B cell, T cell that belong to adaptive immunity. It uses the perforin granzyme to induce the apoptosis of virally infected cells and tumor cells. You guys know in apoptosis there will be interferon stimulation by where there will be cytochrome C will be released and apoptosis will be done. And there will be activity enhanced by interleukin 2, 12, interferon alpha and beta, and produce interferon gamma that will activate the macrophages. And it induced to kill when exposed to non-specific activation signal. When it is going to stimulate, when it is getting any non-specific activation signal, but it is also having inhibitory signals that are MHC I on target cell surface. Whenever the MHC class 1 is present on target cell surface, then it will be inhibited. Also killed via antibody-dependent cell-mediated cytotoxicity. What is antibody-dependent cell-mediated cytotoxicity? It means it needs an antibody to get activated. So the CD16 of natural killer cells go on to attach with immunoglobulin G, Fc region of immunoglobulin G, and it will be activated. Same like it is happening with eosinophils. Eosinophils also needs IgE to get activated. Okay, there are two cells that are going to activate antibody-dependent cell-mediated cytotoxicity, that are natural killer cells that need IgG that will attach with CD16, and second eosinophils that needed IgE. And the natural killer cells also having some explanations like how that markers are going and enhancing the thing. And thirdly, there is one more marker present in natural killer cells that are CD56. The CD56 also is called NCAM. The NCAM means neural cell adhesion molecule, and it expressed on the surface of natural killer cell. It is a useful marker. It is a useful marker for the natural killer cells. Secondly, it is these CD56 are going to found in brain and neuromuscular junctions. And secondly, is aids in binding to other cells. You already done with antibody-dependent cytotoxicity, in which there is activation of natural killer cells by IgG, and also there is activation of eosinophils by IgA. Secondly, MHC class 1 is the surface molecule of most of human cells, and it's present antigens to CD8, or you guys know CD8 T cells, and it activates the adaptive immunity against the intracellular pathogens. Some viruses are going, and you guys know the CD8 and MHC are going to attack against the viruses, and some viruses is going to block the MHC class 1. When the natural killer cell find any cell having decreased the expression of MHC class 1, they go and kill that cell and destroy that cell. Why? The natural killer cell suspect that in that cell the virus is going to block MHC class 1 expressions. Therefore, they think that in that cell the viruses are going to grow. So they go and kill and destroy that. And simple, easy. At the natural killer cells, you guys know having the same lineage as a T cell and B cell, having which lymphocyte, and they don't mature in thymus. They're having no memory, and they don't require any antigen presentation by MHC.